You've probably heard of viruses before. They're little infectious agents that can insert their own genetic material into the host cell's DNA and trick it into creating viral proteins to make up new viruses. However, have you ever heard of retroviruses? These are a special group of viruses that can convert their RNA genetic material into DNA through a process called reverse transcription. In fact, HIV is a retrovirus that utilizes this process. Integration of the HIV DNA into the cell's DNA is ultimately what causes the cell to commit suicide. Without reverse transcription, integration wouldn't happen, and neither would cell suicide.
As a result, to save lives, it is important to understand how the process of reverse transcription actually works.
Okay, so before we talk about the process, it will be helpful to know the basic genetic structure of a retrovirus. It is composed of RNA or ribonucleic acid. The coding region codes for proteins that make up the virus. The LTR are identical and each has a U3, R, and U5 region. The R regions are important to reverse transcription, and the primer binding site and polypurine tract are as well, because they provide areas for primers to stick onto. Essentially, a primer is a sequence of DNA or RNA that acts as a base for nucleotides to build off of in the synthesis of new DNA strands. Okay, so now on to the actual process of reverse transcription. In the beginning, the RNA starts out without sections of the LTR. In this animation, the strands that glow red are RNA, and the strands that glow blue are DNA. So basically how it works is a tRNA acts as a primer and binds to the PBS, and a very important enzyme called reverse transcriptase that is found in the virus enables the entire process and attaches and extends the strand. The reverse transcriptase enzyme also degrades the strands. So just watch as the strands kind of reshuffle around and get degraded. Now both strands are extended. And now you have a full double-stranded viral DNA.
Through understanding reverse transcription, scientists have developed medications to block certain parts of the process. Most of these target the reverse transcriptase enzyme, which, as you saw in the animation, enables the entire reverse transcription to occur. Use of these medications in antiretroviral therapy has decreased death rates due to HIV and AIDS related diseases by 50% since 2005. Consequently, the discovery of how reverse transcription works is a breakthrough that continues to save millions of lives around the world each year. This is Annabella Lee, and thank you for listening.